Hi, my name is Philip Calhoun. I'm an undergraduate researcher at the College of Veterinary Medicine at Michigan State University in the JRavi Lab. My lab is focused on creating a computational approach to identify unique pathogenic sRNA signatures in human and non-human hosts. So what's wrong with the current diagnostic methods? The current diagnostic methods have the issue achieving accurate and sensitive diagnosis, which is often confined by closely related or environmental bacteria, or the diagnostic expert might not be familiar with the pathogen or its symptoms. So achieving an accurate and time-sensitive diagnosis is important because it gives, if it's undiagnosed, it gives pathogen time to spread across populations, and if left untreated, fatalities occur for numerous hosts. And also, if the pathogen is zoonotic or if it's animal pathogen, it's expensive to treat a whole herd for a certain pathogen. So my objective was to develop a general computational workflow to identify pathogenic sRNA in affected host samples, and to generalize the approach so it could be applied to any pathogen of interest. So I mentioned that we're using sRNA, and sRNA are small, important biomolecules in gene expression. We'll be looking at mRNA in particular. And when a pathogen infects a host, changes occur in sRNA expression, and it becomes possible to detect bacterial sRNA alongside host and host non-coding RNA in the host sera. It is also stable in numerous pHs and stable over long periods of time. So for the computational workflow, using a Mycobacterium avian paratuberculosis infection within the cow to then generate and test our workflow. So first you have to sample the host sample sera using dual RNA-seq from infected host samples. Then you perform trimming and quality control. The next steps are done in concert. You find mRNA databases for host and pathogen and identify host pathogen reference genomes, which plays into the alignment step when you align the sequences against mRNA, sRNA databases and reference genomes. Then you have to quantify the overall differential expression of the reads across conditions such as time. So for the alignment data, since we have a Mycobacterium avian paratuberculosis infection within the cow, we should see high amounts of assigned reads, assigned and mapped reads within the map infected, and then we should also see high assigned reads within the within our host with a control and infected, which is the cow, which we do see. Right here we have a differential expression log2 heat map for both cow on the left and map on the right. Since it is log2, a slight difference in color signals an exponential change in overall expression. So a good target diagnostic molecule will have a high expression in infected or a dark color and a low expression rate in the light tiles, which I also identified two, which could be potential diagnostic target molecules within a map infection within the cow. Querying against host pathogen reference genomes with suitable controls helps identify sRNA unique to pathogens of interest, and the higher magnitude differential expression for specific sRNAs in affected samples makes for better diagnostic target. Higher match reads with map infected could lead to further identification of map sRNA within the infected sample. And sRNAs are not identified with reference genome database searches will be identified using de novo prediction. Differential expression of sRNA could help target early diagnostic biomarkers and detect host mRNA changes. We have map RS12475, which was the top black mark. That's the products for ribosomal RNA within MAP, and then also MAP RS19585, which was the black mark, codes for the tRNA glycine. So for future direction of the project, we need to benchmark against other existing sRNA discovery pipelines. This means that we're just going to test our workflow against other workflows that have already been determined as correct. And then we have de novo methods to predict other novel mRNA. And this basically means within the sample, we're going to use tools to then have ways to label the other unknown miRNA as host or pathogen. Then we have miRNA target and pathogen prediction. This is basically telling us what what the mRNA does within the pathogen and how it affects its overall development. I'd like to acknowledge Nora Sasinski, Samuel Chen, Karn John Naranson, CVM and MSU, Brush, Dr. Ewert, and my NIH grant. Thank you for your time. If you have any questions, please use the questioning service, and I can be contacted through the avenues below. Thank you, and have a good day.